Hello, and welcome to my series of videos describing the Kinomics toolbox. Before we begin actually exploring the toolbox, I would like to define Kinomics and describe the platform we utilize to study the kinome. Kinomics refers to the study of cellular kinases and their targets. Kinases are enzymes that major function is the transfer of phosphate groups to targets to either propagate a signal or cause a direct effect. They are involved in nearly every cellular pathway, from cell growth and replication to circadian rhythm. There are a number of methods that can be applied to study the kinome. However, here we will discuss the PamGene PepChip platform. The PamGene PepChip platform is a peptide microarray with 144 or more residues, each representing a known intracellular kinase target. Each target is a peptide composed of approximately 15 amino acids, where one or more of those amino acids are phosphorylatable residues, such as a tyrosine, a serine, or a threonine. The general idea is that a specially prepared cellular lysate is added to each well of the chip and allowed to act on the kinase targets present in the microarray. As the kinases act on the targets, specialized fluorescent antibodies bind the phosphorylated residues, allowing the phosphorylation to be quantified as a measure of fluorescence. In other words, for an image of an array like this, a bright spot, such as this one, has had more phosphorylation occur than a lighter spot, such as this one. If we take a look at the array itself close up, we can see that it is a porous structure. This is the array under an electron microscope, and it's a porous, three-dimensional structure where each of the pores is lined by a specific peptide. Each one of these peptides, as we've mentioned, has at least one residue that can be phosphorylated. The general idea here is that the sample sits on top of the microarray, and then as it passes through the microarray, the cellular kinases are allowed to act on these targets. Once it's passed all the way through, the cycle repeats itself by being pulled back up through the array, once again acting on those targets. This pumping action allows for an interesting type of data to be captured. Essentially, what we can do is allow the sample to sit at the bottom of the microarray, and then we can take a picture of the array itself, and then we can continue the pumping action, and then take another picture when it's returned to the bottom. This gives us an idea of kinase activity over time, or a measure of kinase kinetics. Another way we can look at this data is by taking multiple pictures of the array in the same state. So this is an example of varying the exposure time of the camera. If I take a picture for only 10 milliseconds, I get this much phosphorylation as measured by the fluorescence of a given spot. If I increase that to 20 milliseconds, 50, 100, 200, I get a linear response with increasing signal. The other thing to mention here is that there is a foreground and a background signal. The foreground signal is a measure of the fluorescence of the spot itself, and the background signal is calculated by measuring the fluorescence of four corners in a square grid around the spots themselves. Finally, just to give you an idea of what this looks like overall for kinetic data, here is a single spot measured from cycle 32 to 92. You can see that it's increasing in fluorescence as time goes on. And you can see when we plot this change in fluorescence, it shows a linear phase with a slight slowdown as time goes on. This is an idea of what all the kinetic data ends up looking like, with some complications. Thank you for watching this video explaining Kinomics. The next video will focus on the base level data and how it's visualized in the Kinomics toolbox.